unfold in real time. Just a couple of disclosures. You heard a little bit about family history tools. I'm not going to talk about that right now, but I'd love to discuss it more during the break. And we're going to talk about how elephants are helping people. And if I do my job right in the next 19 minutes, you, like me, will be a lover of all things elephant, if you're not already. What are we going to do? Well, we're going to talk about elephants. Why elephants? Why are we studying these elephants? We're going to learn about elephant P53. How is that different or the same as human P53? And then the most exciting part to me at the end is how are we translating this to people who may be sitting right here in the room one day in the future? I always want to start off, though, with the patients. We heard a really moving testimony this morning from many different patients. This is Lauren, and Lauren has got Li-Fraumeni syndrome. She's working on her third cancer now, but I love this picture that her mom sent to me because of the henna tattoo of the elephant. This is the wrist of a patient, Abby, who came to visit us in Salt Lake. She had this bump that was growing. She had Li-Fraumeni syndrome. She's 8. She had a choroid plexus carcinoma when she was younger. This turned out to be, unfortunately, an extra-osseous osteosarcoma, soft tissue sarcoma. Here she is visiting the lab with her brother, who also has Li-Fraumeni syndrome, who's also had cancer when he's a young age. And the thing about that tumor that I want to point out is, this had genomic profiling and sequencing, and the only mutation, the only genetic change they found in Abby's tumor was that it was missing P53. So P53 is very, as you guys know, very important and may hold some really intriguing hope in terms of therapeutics. And then the youth workshop. These teenagers, Jen mentioned that this was life-changing experience for all of us that was there. And it was amazing to bring all the teenagers together. I do want to point out one particular teenager. This is Addie. Now, Addie I met, and she unfortunately passed away several months ago. And I've not done this before in a lecture, a public lecture, but I want to dedicate this talk to Addie. And her mom... Her mother is here, and we were talking last night, and what would Addie like the most? Addie loved to be in the limelight. So I hope that you'll think of Addie and everything that we're doing and all the speakers. It's for everyone here, but especially my talk is for Addie, because we miss her dearly. Okay, so elephants. Elephants rarely, what's the deal with elephants? Why am I always talking about elephants, and what do elephants have to do with P53? Well, first, just a little bit of biology. Elephants, it turns out, rarely get cancer. Okay, well, that's interesting, but why do we care? Well, if you look at an elephant is 100 times the size of a person. That means they have 100 times as many cells. So people have roughly 37 trillion cells in their body, give or take a couple trillion, right? So that means that elephants, I just did the math on Google, have 3.7 quadrillion cells. That's a lot of cells. Elephants live 50 or 60 years. That many cells dividing decade after decade after decade, just by chance alone, all elephants should be dropping dead from cancer. But they don't. Because if they did, there'd be no such thing as elephants. So elephants had to evolve some way to naturally protect themselves from developing cancer. It turns out a really smart guy and his student, Carlo Meli, looked at the genome of the African elephant several years ago. And I was at a conference, and he was explaining that elephants, instead of 2 copies of P53, have evolved 40 copies. So 20 times as many. I almost fell out of my seat when I heard that. But then he went on to say he wasn't sure if that was the real reason they weren't getting cancer, but it was just an interesting observation in the genome. So I went right up to him afterwards, and I explained, I said, Dr. Meli, I'm taking care of all these wonderful, amazing patients with Li-Fraumeni syndrome. They're missing their P53, and they're almost always getting cancer. And you said elephants with the extra P53 almost never get cancer. What if somehow, some way, we could get some elephant blood, study it in the lab, right next to the Li-Fraumeni blood, and really try to understand if this is the reason elephants don't get cancer? And it kind of, for me, flipped the paradigm of the cancer research we were doing in the lab. Instead of looking at who's getting more cancer and why, like the patients with Li-Fraumeni syndrome, What if we looked at who wasn't getting cancer and how, and try to emulate that? And this led to a lot of publication and fanfare. It was published in the cover of Newsweek. This was a very interesting experience for me because people would come up to me at scientific meetings and say, hey, you're the doctor who published that article in Newsweek, right? And I said, no, I don't write for Newsweek. We published in JAMA. That's with the referees, the peer-reviewed. That's what I'm proud of. But one of the nice things about the Newsweek and the publicity is every story that was published around the world on this, I made sure to mention Li-Fraumeni syndrome. And Jen spoke about awareness, and in this Newsweek there was actually a family that was highlighted at the zoo with Li-Fraumeni syndrome. The father unfortunately has passed away since then. But for me, what made me most proud, although the JAMA was great, it was actually being able to help spread the word about Li-Fraumeni syndrome. So how... Oh, well, you don't have to, it takes up my minutes. I can't have you clapping. Save it for the end. Okay, so how is it working? Well, elephants have these extra copies of P53, and if I can turn on the laser without poking out my eyes, they have one copy, and it looks like it's not working, so we'll use the mouse. Elephants have one copy, so two copies, but one version of P53 that's very similar to the human P53, it's full length. But the other 19 is very interesting. These are called retrogenes. Okay? It doesn't mean they're from the 70s or wearing bell-bottom pants or anything like that. These retrogenes, also known as pseudogenes, are smaller genes. But you can see that they actually have some components of the DNA binding domain where the DNA links, but not entirely. And interestingly, they don't have that part of the P53 gene that allows it to form the tetramers. But they're definitely there, and working with people in the lab, we said, well, let's focus on the wild type, and let's focus on this one called Retro9. Retro9 is very similar to, or I should say, in the elephant, it's the gene that's most expressed. So we said, let's just pick that one and start working with it. Dr. Beglin, who's here at the conference, did a lot of work, and we showed that the elephant P53, indeed, it triggers cell death, and it seems to work in the elephant cell even better than a human cell. So in other words, if you look at a human cell and you expose it to radiation or stress, then most of the time the cell will stop dividing, and usually it will apoptose or undergo cell death, so it doesn't go on to turn into cancer. Simplifying all of what we talked about this morning, but just keep that in mind, okay? But an elephant cell, if you even look at an elephant cell funny, if you zap it with radiation or stress it, it's so sensitive it undergoes very rapid apoptosis. Okay? And that makes sense because the elephants can't develop cancer. There'd be no such thing as elephants. Well, since that publication has come out, we've worked with some in the room, who I'll mention later. We've been studying really hard. How is that elephant cell different, elephant P53, different than the human P53? And you can go through and you can see what areas are similar, what areas are different. I'm going to breeze through all of these experiments just so you get a flavor, a taste of what we're doing. We're able to actually go through and we can mutate different parts of the elephant gene and see how does the elephant gene perform, and then we can go and we can compare it and see which amino acids are the same, which are different, and then transfer those into a dish and keep growing them. So we're trying to understand what are the parts of the elephant P53 gene that are working that give it its special ability. One thing that we did is we said early on, listen, you know, I'm a pediatric oncologist. I take care of patients with Li-Fraumeni syndrome. We want to think about translating this to patients. So what happens when you put an elephant gene, an elephant P53 gene, into a human cancer cell? And we worked a lot with osteosarcoma. We know this is a big cancer that our patients get. So we can take GFP, which is just a marker and lets you look at the cell, and you can put GFP into an osteosarcoma cell, and you can see that the cell has some amount of death. This basically is a measure of how much of the cell is dying. Over here, this Western blot on the end, this basically just shows you the protein is made. Well, then we could put in one of the smaller genes, the retro gene, and we see actually, you know what? Even though it's small, it's causing some amount of cell death, and it's made into a protein. When we put in the wild type or the normal elephant P53 gene, the full-length one, it creates a lot of cell death. So that was nice to see. Dr. Abeglen has done a lot of work, and she has shown that, you know, it looks like molecule per molecule, preliminarily, the elephant P53 seems to work a little better than the human P53. And what's really interesting, here you can see these are just osteosarcoma cells, and they're growing here, sort of wide, healthy cancer cells, if you will. But if we put in the retro gene, you see they're kind of smaller. If we put in the ancestral gene, more of them die, they're even smaller. But when you combine the two, because remember, in the elephant, you have all of the p53 genes working together, you see, well, see, there's the camera. You see much more cell death, right? So there seems to be an interaction between the two types of p53 genes. And this is work done by Dr. Goldfeder, who came all the way from Israel, who's sitting in the audience. When we do these gel shift assays or DNA binding, when you actually put the two proteins together, so Moore is able to actually synthesize elephant p53 in a really elegant way. When she puts it all together, the elephant retrogene, the smaller one, and she mixes it with the wild-type elephant p53, that wild-type elephant p53 is binding less DNA than it usually does. And it also is the same, that's where you see the curve sort of go down, and the same is true when you add it to human p53. So there seems to be this biological, physical interaction, which we're now trying to understand more, between the two different types of elephant p53. Now, when we look to see some of these target genes, right, the transcription, what happens with elephant p53, it actually activates some of the same genes as the human p53 when it's full length, and that's not a surprise. CDK, N1A, BAX, and MDM2. We heard about MDM2 earlier. But what's interesting is the retro gene doesn't really seem to activate the same transcriptional program. And in fact, there's really only one gene that seems to be targeted. And so where does EP53 go? What is it doing if it's not doing all of the transcription? Well, it's in the nucleus. We know that, the ancestral. But we also know that the GFP, when we tag it to the retro 9, that's so we can see it, that tends to be more in the cytoplasm. So they're in different locations. And we're actually able to divide the cell up and see where in the cell is it actually working. We look at the different cellular compartments. And what seems interesting is over time, when you mix the two together, it's almost as if the retro gene, the small elephant p53, seems to be pulling, in a sense, shifting the ancestral p53 into the cytoplasm. So we're really excited by this, and we're trying to understand, how is this working? Is there transcription? Is it non-transcription? And so these are all the things that we're concentrating on. How am I doing for time? 5 minutes. Great. Okay, so here's just more evidence of trying to figure out where it works. Okay, so now we get to the fun part in the last 5 minutes. So we said, listen, we need to figure out a way to try to get this to people. What can we learn from this elephant p53 to actually help people? So we actually worked with the University of Utah. We worked with our collaborators, Technion, the Israel Institute of Technology, right? That's like the MIT of Israel, or they like to say MIT is like the Technion of the US. Okay, so those are still awake. But anyways, so why peel therapy? Well, peel is the Hebrew word for elephant. And so working together, we said, let's see if we can make a new elephant-based medicine. What I'll tell you is that Peel was a little bit nervous about me showing you the data I'm going to show them, but I assured them that you're going to keep it confidential, and it was too exciting not to show. And it's all very, very early and very preliminarily. All right, data. So we'll see what we think about it during the break. Okay. So we take a mouse and we give the mouse breast cancer. We give the mouse mouse breast cancer, and we put in we have a nanoparticle, right? And so Peel has all these proprietary delivery systems and combinations of the elephant-based medicine. And so you give a placebo, and what happens over time? Well, the breast cancer grows, because that's the placebo. But then you deliver this new medicine that they're working on based on this elephant P53. And what happens after 2 doses? Well, it seems to level out. Here's just another view of the cancer in the mouse, and you give the drug, and after a week, the tumor, it pretty much begins to shrink, right? So that's kind of cool. And you can actually look, and you can see the size of the tumor with the placebo, and you give this new elephant-based medicine. Again, it's very, very early, but encouraging, much smaller tumors. And then in Utah, what we're able to do is we do these nice 3D images of mice, right? This is a single mouse with multiple images. And we have osteosarcoma from patients with Li-Fraumeni syndrome that we take out. So this is, see where the arrow is? That big red fireball? That's an osteosarcoma. That's a human osteosarcoma in a mouse, which we're going to treat with elephant P53 medicine. It's a bit like Noah's Ark, okay? So this gets the empty one. Nothing happens 1 day later. But look, when we give actually the new medicine that we're working on with this company, Peel, well, in 24 hours, we see an over 50% reduction in metabolic activity of the tumor. So that's very encouraging. We looked at Ewing sarcoma, a cousin of osteosarcoma. Doesn't occur that often in Li-Fraumeni syndrome, but this is an important cell line because it's missing P53. And what happens after we give a dose? We get, again, 50% reduction. Okay, so that's nice to see. Now, this is just metabolic activity. What you really want to know is not how much is the cell dividing or taking up glucose, but are the cells actually dying? And what we see is that in the one with the control, we have a baseline amount of tumor death or necrosis. That just goes on as a tumor grows quickly. But we saw over 90%, and that's a clinical pathologist telling me that, not my own judgment, but I was happy to see it, that we have this tumor that's really being wiped out by, again, this elephant-based medicine. And then the last example before my time goes up and my little wristwatch starts zapping me, I'm guessing. So here is a canine osteosarcoma, a very aggressive bone tumor, same osteosarcoma that people get. And you can see you give the empty nanoparticle and really no change, okay? And again, the tumor is the bright yellow-red, but then we give the medicine and we get an 80% reduction in metabolic activity in 24 hours, okay? Now, this is very, very early. Here's a close-up. You can see it. You know, the tumor, it's not that it melts away, it's just that the metabolic activity stops working. And again, the reason Peel was very nervous about me showing this, but I said, I gotta show it to the Li-Fraumeni folks, this is so important, is because we don't know if this is gonna work, right? It's super, super early. It's preclinical. We know just because it's preclinical doesn't mean it's going to make it to clinical. You're going to hear more about clinical drugs in the next couple of days. But we're excited to keep working on it. We got to make sure it's safe. We've got to make sure, importantly, if we're giving this to our kids, right, are there side effects that we're not considering if you give an elephant medicine? So these are things that we're working on. So that, by the way, that's Dr. Schroeder, and that's me, and that's an elephant. And it's always very important to say I'm making that face not because when I'm stepping in, but because the sun is in my eyes. So at the end of the day, why elephants? Well, elephants are naturally protected from cancer, right? Evolution has figured out how to modify, tinker with P53, we think, to make it even better than human P53. We're studying it with many people to try to understand exactly how it's working, and you heard how we're working to translate it. So many people to thank as my time is up. I do want to point out this EP53 amazing team, David Malkin, who's been a wonderful mentor and friend to me throughout the years, David Lane, Carol Preeves, Laura Attardi, everyone working together, and here is the lab. And basically, the idea is, what can we learn from elephants? Well, they've had 55 million years to learn how to avoid cancer, so now we're trying to learn from them to help everyone here in this room. Thank you. Okay.
Thanks, Jacques. You have for a question?
Yeah, about it.
It's a very beautiful talk, and we have a lot of expectations from the team.
Yeah.
But my question is, your family of genes, are they actually alternative splice form as those which we know in the human?
Right.
Or are they, maybe some of them at least, are pseudogenes, which are
Yes.
back incorporated in the genome, and this is maybe why they're called retro, like retroviruses?
Yes, like retrovirus. So Varda's question was basically, what's going on with these retrogenes? How are they getting put back into the genome? And are they really pseudogenes or not? So just, again, a 1-second biology lesson. We know that usually things go from DNA to RNA to protein. What we believe that these genes have done way, way back, millions of years ago, is they went from DNA to RNA and then back to DNA because they're missing introns. We don't think all of them are expressed at the same level, but both Asian elephants and African elephants have all of these, so we believe they've been under positive selection. throughout evolution, and we're actually getting ready to do some RNA-seq experiments to really try to understand. We're studying simultaneously elephant p53 in human cancer cells, but also in the native elephant cell environment, because we know that each of the retro genes are expressed at different levels, and that may be a reflection of their importance. And I'm really excited hearing Dr. Lane's talk this morning about some of the experiments we could do to try to maybe actually tease that out a little better.
A small sub-comment is, do they share the same promoter? Because David mentioned we didn't
start the promoters. Yeah, we're trying to answer that question now. So to be continued.
Okay, one more from Arnie, and then we'll have to move on.
So you started off with this Peto's paradox, which suggests that the elephant has a lot more cells than us. And that's certainly true. It's only 10 generations more than us, though. Mm-hmm. So it's 2 to the 10th more cells. And 2 to the 10th cells are the number of T cells we have in our body, right? So it can't be that the elephant is at higher risk just because it has 10 generations more division. And it must turn over a lot of cells over its lifetime. Absolutely. Like we do.
Yes.
So the argument that cell number is really a critical number here in terms of scale is probably not a valid argument, just from the numbers that we have.
Right.
But secondly, there's a group in China that actually have an approved product that is in a virus with a human p53 put back into head and neck tumors. And there's a sort of interesting observation that they've made that's different than yours. And that, I think, would be an interesting thing to follow up. It does not actually reduce the tumor in humans until you exirradiate it.
Right.
And that's because most of the time p53 gives rise to senescent phenotypes rather than apoptosis. Yeah. And so that may be a difference that could be interesting. But we do know the human equivalent of p53 going into cells will kill those cells as well. The problem is they don't discriminate between normal cells and
right. And so I think this gets much longer for the time I've got to answer that, but I would say the elephants, what's very interesting is, it is more than just the number of cells. And biologically, in a dish, there's a very obvious difference in the way that they respond to increased radiation. And that p53 in the elephant, we think, it actually turns more towards apoptosis than cell cycle arrest. So we think there may be something a little bit unique about the elephant, or a lot unique about the elephant p53, which may be why we're seeing some of this necrosis. But again, it's very early, but certainly it gives me career stability for at least the next 5 to 10 years. So thanks very much.
Thanks, John.